So, with reference to the cancer drug discovery and development, today we will discuss on inflammation and how it is involved in the initiation of the cancer, and inflammation, the various factors that are involved in inflammation can be taken as targets for drug development. So, I was mentioning that the tumor microenvironment, the moment a normal cell gets converted into a neoplastic cells, infiltration of the immune cells takes place, and this infiltration of the immune cells leads to the supply of various growth factors that are required for the cancers, and the proliferative signaling, survival factors, angiogenic factors, matrix modifying enzymes, which will facilitate angiogenesis, invasion and metastasis, and induction signals leading to the activation of epithelial mesenchymal transition takes place. And all these signals which are mediated by the immune cells leads to the sometimes foster the development of pre-malignant to malignant cancers. And these inflammatory cells facilitate the release of chemicals, particularly reactive species, which are again mutagenic and hasten the malignant process. So, to give a definition on inflammation, it is a cellular, immune and metabolic response to injury or infection. It is a protective vascular connective tissue reaction, which is designed to eliminate the cause of injury and also clean up the debris that is associated with dying cells and tissues. And the various hallmarks of inflammation are increased temperature, redness, swelling, pain, loss of function. And the mediators of inflammation, it is a complex interplay of cellular and particulate matter. The chemokines, plasma enzyme mediators, lipid mediators and cytokines, these are the major mediators. Among these, the lipid mediators are the most important signals which are involved in mediating inflammation. In fact, inflammation has two phases, the good side and the bad side. Inflammatory response to tissue damage is of great value, which helps in isolating the damaged area and mobilizes the effector cells and molecules to the site and promoting the healing process. But when this inflammation becomes chronic, then that is the one which leads to and causes more damage to the body than the agent itself. And in this process, the various examples of inflammatory disorders include Alzheimer's disease, anaphylaxis, asthma, atherosclerosis, atopic dermatitis, chronic obstructive pulmonary disease, Crohn's disease, gout, and a number of other disorders like arthritis and psoriasis. A number of other disorders are there. The list goes on with reference to inflammatory disorders, including the cancer. The connection between inflammation and cancer, it is a critical component, as I was mentioning earlier, in tumor initiation and progression. Cancer can arise from sites of infection, chronic irritation and inflammation. The vital component of the tumor microenvironment is the inflammatory cells, which are infiltrated to the site of cancer cell. And this inflammation promotes the neoplastic process involving proliferation, survival and migration. And tumor cells have co-opted inflammatory signaling molecules and receptors. In fact, we can say that tumors act as wounds that fail to heal. And keeping all these components, in fact, the Time magazine has come up with inflammation as a secret killer, which is involved in promoting various disorders including cancer. So, chronic inflammation is a risk factor of cancer, which is exemplified in case of ulcerative colitis, where the chronic inflammation leads to dysplasia and then adenocarcinoma. The patients with ulcerative colitis have a 5 to 7 times more chances of getting the colon cancer. In fact, the colon cancer persists, if it persists for 30 to 40 years, then it increases the risk by 20 to 35%. And colon cancer associated with inflammatory bowel disease has the worst prognosis. When they both combine, then the cancer prognosis is very severe. And management with the anti-inflammatory agents was shown to reduce the cancer. So, these are the evidences which clearly tell about the role of inflammation in cancer. Chronic inflammation in terms of the various factors which is explained in this figure. The insulated stromal cells recruit activated inflammatory cells. The cancer cells will recruit these stromal cells to generate various growth factors, and these inflammatory cells express factors that stimulate cell growth and progression. And as you can see from the mutations which are involved in response to variety of physical, chemical, physical and biological factors, convert normal cell into neoplastic cell. So, when epithelial cells get transformed into initiated epithelium, and the promotion of this initiation process is mainly done by the inflammatory cells which express the various growth factors and other cytokines. And as long as it is confined to a particular location, it is called as the benign carcinoma. But because of the inflammation, it helps in migrating to other sites, which is the malignant conversion. So, inflammation is involved not only in the promotion of the initiated epithelium, cancer epithelium into benign, and also in invasion and metastasis. Examples of various cancers in which inflammation plays an important role is inflammatory bowel disease, which results in colorectal cancer, gastric intestinal metaplasia, where gastric cancers are associated, Barrett's esophagus is associated with esophageal cancers, chronic hepatitis leads to hepatocellular carcinoma, chronic pancreatitis leads to pancreatic cancer, oral leukemia leads to head and neck cancers, ductal carcinoma in situ leads to breast cancer, bladder dysplasia leads to bladder cancer, and cervical dysplasia leads to cervical cancer. So, as you can see, inflammation associated with various tissues is ultimately leading to various cancers, and that is why inflammation has been designated as the 7th hallmark of cancer. So, inflammation is a complex pathway and promises to be more complex, and it is associated with many disorders including cancer. And that is why anti-inflammatory drugs, non-steroidal anti-inflammatory drugs have become our favorite medicines. Humans have been consuming non-steroidal anti-inflammatory drugs for more than 3,500 years, and each year approximately we are consuming around 40,000 metric tons of aspirin, that is 120 billion aspirin tablets of 300 milligrams in size. So, you can imagine the role of inflammation in various disorders. After 3,500 years, the first real progress in our understanding of the mechanism of non-steroidal anti-inflammatory drugs action came about 40 years ago, when it was shown that all these anti-inflammatory drugs like aspirin, indomethacin, ibuprofen inhibit the prostaglandin biosynthesis, and these prostaglandins are the bioactive lipids which are formed from polyunsaturated fatty acids like arachidonic acid and then generated, and all such kind of oxylipids which are formed from polyunsaturated fatty acids like arachidonic acid are termed as eicosanoids, which include the prostaglandins, which are unrelated compounds like leukotrienes are included under eicosanoids. most of these eicosanoids are produced from arachidonic acid by oxygenation pathway, and they are termed as the local hormones or autocoids, and they are rapidly degraded from the site that they are produced, and that means that they have very limited half-life from the thing. In addition to participating in intracellular signaling, they are also involved in intracellular signal cascades. The main steps which are involved in eicosanoid biosynthesis is first, activation of the phospholipase A2. So, the membrane bound phospholipids contain unsaturated fatty acids. The activation of the phospholipase A2 is the first step which releases the fatty acid from the membrane bound phospholipases. Once this is released, then synthesis of these oxylipids by various oxygenases takes place. The key oxygenase pathways include COX and LOX pathway and subsequent cell specific modifications leading to various other factors. So, to give more information about the phospholipase A2 activation, as you can see from the figure, the membrane phospholipids, any kind of internal signal or external signal leads to activation of the phospholipase C, and phospholipase C in turn activates the release of calcium from endoplasmic reticular stores or other internal stores, and calcium in turn activates the various kinases, and these kinases in turn leads to activation of phospholipase A2, which in turn releases arachidonic acid. So, as you can see, the first step, activation of the phospholipases, leads to the release of fatty acid from membrane bound phospholipids, and the key enzyme in this is phospholipase A2, which acts on the second carbon of the glycerol backbone of the triacylglycerol and leads to release of the unsaturated fatty acid. And the third step is eicosanoid biosynthesis, which occurs in almost all cell types except the red blood cells, and there are three pathways that are associated. The cyclooxygenase pathway, which incorporates two molecules of oxygen and produces the prostaglandins and thromboxane. The lipoxygenase pathway produces leukotrienes, lipoxins and hydroxy metabolites by incorporating one molecule of oxygen. The third pathway includes the cytochrome P450s or monoxygenases, which produce the various hydroxy metabolites of the fatty acid. So, these are the three pathways where incorporation of the molecular oxygen leads to the various oxylipids by incorporating either one atom of oxygen or one molecule of oxygen or two molecules of oxygen to give rise to various bioactive lipids. And this cyclooxygenase pathway mediated signaling, as you can see from this figure, that phospholipids from phospholipases gets activated in response to either physiological or pathological stimuli. Release of the fatty acid takes place, then the prostaglandin synthase or cyclooxygenase acts on this to produce an intermediary molecule which is called as prostaglandin H2, and this prostaglandin H2 forms a substrate or precursor for various tissue specific cells, and they are involved in the release of various other molecules. For example, in case of the platelets, prostaglandin H2 is converted to thromboxane by the thromboxane synthase pathway, whereas in case of the blood vessel walls, the same prostaglandin H2 is converted to prostacyclin. And in all inflammatory cells, wherever there is infection that is taking place, leads to the conversion of prostaglandin H2 to prostaglandin E2, and this is the most important inflammatory mediator, and this in turn acts through the G protein coupled receptors or GPCRs, which are expressed in different cell types, and there are 4 different types of GPCRs for prostaglandin E2, and which are involved in mediating all the cardinal symptoms of inflammation like pain, fever, redness, swelling. All such kind of symptoms are mediated by the prostaglandin E2, which is formed from arachidonic acid, which is released from the membrane phospholipids after action by the cyclooxygenase pathway. And there are two isoforms of this cyclooxygenase, cyclooxygenase 1 and cyclooxygenase 2. And cyclooxygenase both are involved in oxygenation of the arachidonic acid and then produces the prostaglandins, whereas while the COX-1 is constitutive, it is always present in the normal cell. Physiological prostaglandins are produced and they are involved in the normal process like blood clotting, kidney function and stomach protection, gastrointestinal protection. Whereas the COX-2, which is induced in response to inflammatory stimuli, produces the prostaglandins which mediate the pain, heat, swelling, which are the cardinal symptoms of inflammation. So, the COX-2 is the major mediator of inflammation, and this COX-2 was shown to be involved in the correlates very well with the initiation and progression of the cancer. As I mentioned earlier, COX-2 is not present in the normal cells, but it gets induced in response to variety of stimuli. And as the normal cell gets transformed into a cancerous cell, neoplastic transformation, you will see the COX-2 initiation, COX-2 induction. And the extent of COX-2 is correlated well with the progression of the cancer, normal epithelium to carcinoma. You will see increased expression of COX-2 and it correlates very well with the cancers. And COX-2 expression has been shown in variety of cancers like breast cancer, cervical cancer, prostate cancer, bladder cancer, colon and stomach cancers, head and neck cancers, liver, pancreas, bile duct, skin. So, in all such kind of cancerous tissues, there is overexpression of COX-2, and this clearly tells that COX-2 can become a target for the development of anti-cancer drugs. And as I mentioned earlier, non-steroidal anti-inflammatory drugs have been in the market for the past several centuries, and which include ibuprofen, aspirin, diclofenac, naproxen, indomethacin and all these kind of things. And they are involved in action, inhibition of both COX-1 as well as COX-2. And as I told earlier, that COX-1 is constitutive and it is cytoprotective. And conventional anti-inflammatory drugs inhibit both COX-1 as well as COX-2. Inhibition of COX-1 leads to various side effects like gastric irritation that you see and kidney failure, whereas inhibition of the COX-2 gives you the relief from all inflammatory symptoms. And that the discovery of COX-2 as the inducible isoform mediating inflammation led to the development of COX-2 specific anti-inflammatory drugs. And in fact, both the structure guided information was taken for developing the COX-2 specific drugs. Both COX-1 and COX-2 crystal structures are available, and the only difference between COX-1 and COX-2 is the COX-2 site, active site is much bigger than the COX-1, which has been exploited for the development of COX-2 specific drugs. And the COX-2 specific inhibitors have come into the market like celecoxib, then rufecoxib, valdecoxib. There are a number of these COX-2 specific inhibitors came into the market, and they are without the gastric side effects which are associated with the conventional non-steroidal anti-inflammatory drugs. And these COXIBs reduce were shown to reduce the colorectal carcinoma polyp burden by 31% in colon cancer patients. And in fact, COX-2 inhibitors have been approved for the treatment of colon cancers with familial adenomatous polyposis patients who are prone for more colon cancers, and they have been up. So, in addition to their role in initiation, progression, and conversion of pre-malignant to malignant cancers, inflammation mediated through the COX-2 has been shown to be involved in multi-drug resistance in cancer patients. What happens is when the multi-drug resistance is the major problem in patients undergoing chemotherapy, the patient responds to the cancer quite well, but the moment after some time he develops resistance, the drug is no more active on these cancer patients, which is called as multi-drug resistance. This was mainly because of one is increased metabolism of the drug that was taken in, or efflux of the drug that enters into the cancer cells, and then various other mechanisms have been attributed. In that, the efflux pumps are the one, and several studies have shown that inflammation mediated by COX-2 leads to increased expression of multi-drug resistance protein, that is peak glycoprotein, and which was shown to be and as a result the anti-inflammatory drugs, specifically COX-2 inhibitors, can downregulate the expression of the peak glycoprotein and thus again overcome the multi-drug resistance. This was clearly shown in case of the colon cancer and even other cancers also. In fact, development of resistance to the imatinib. Imatinib is one of the tyrosine kinase inhibitors in the market for the treatment of chronic myeloid leukemia, and Gleevec is the brand name of imatinib which is there in the market. In fact, Gleevec is the first drug that came into the market on basing on the target based development of anti-cancer drugs, and this specifically targets the tyrosine kinase which is overexpressed in chronic myeloid leukemia patients, and this was also based on the structure based drug discovery. They have come up with Gleevec, but very soon Gleevec resistance was shown. The cancer patients undergoing Gleevec therapy develop resistance and they are no more responding, and this was attributed also from our preliminary studies. We have shown that overexpression of COX-2 leading to multi-drug resistance protein, that is efflux pumps, it was responsible for the development of drug resistance. When you use celecoxib, the COX-2 specific inhibitors, it was clearly shown that it downregulates the expression of COX-2 and thus facilitates the downregulation of the efflux pumps, and thus it helps in overcoming the drug resistance. So far we have studied the cyclooxygenase pathway and its signaling in the mediation of cancers. How the enzymes which are involved in the cyclooxygenase pathway have become the targets for the development of anti-cancer drugs. And in addition to cyclooxygenase pathway, there is another eicosanoid signaling, that is the LOX pathway, and LOX pathway also acts on the substrate fatty acid, that is arachidonic acid. Once arachidonic acid is released from the membrane phospholipids, it is acted upon by the 5-lipoxygenase pathway, and 5-lipoxygenase pathway gives rise to an intermediate which is called as leukotriene A4, and the leukotriene A4 forms the substrate for the various tissues to form tissue specific leukotrienes. By the action of leukotriene A4 hydrolase, leukotriene B4 is formed, and by the action of LTC4 synthesis, leukotriene C4, and then it is further converted to leukotriene D4, E4 and F4. So, the leukotriene C4 and D4 are formed by combination of glutathione to the fatty acid, and that is why they are called as peptidoleukotrienes. And leukotriene B4 on the other hand is non-peptidoleukotriene, and these leukotrienes intermediate the various inflammatory pathways. and as a result the 5-lipoxygenase enzyme, which is involved in the formation of the leukotrienes, has become the target for the development of anti-inflammatory drugs. In fact, gelatin is there in the market as a 5-lipoxygenase inhibitor, and the leukotrienes, similar to prostaglandins, mediate through G protein coupled receptors, and these GPCRs which are expressed in the tissue specific cell types are mediating the effects of the leukotrienes, and as a result leukotrienes receptor antagonists also were developed, and they are also in the market for the treatment of various inflammatory disorders. Gelatin, 5-lipoxygenase inhibitor, and mantelucast, gefirlucast leukotriene receptor antagonists are in the market for the treatment of inflammatory disorders, specifically allergy and asthma. So, this 5-lipoxygenase pathway also plays an important role. As we discussed, the crosstalk between the cancer cells and infiltrating immune cells which are present in the tumor microenvironment is again mediated by the 5-lipoxygenase pathway. Recent reports indicate that 5-lipoxygenase mediates the crosstalk between the infiltrated immune cells and the cancer cells, and the tumor cells with high expression of leukotriene receptors and also 5-lipoxygenase, they promote proliferation, migration and lipogenesis. For example, macrophages and monocytes and eosinophils and other adipocytes and fibroblasts, all these are the cells which are present in the tumor microenvironment. Specifically, the macrophages which are involved in the formation of the leukotrienes, and these leukotrienes have an impact on the cancer cells through the receptor antagonists, and also vascular endothelial cells promoting the angiogenesis and thus metastasis. So, this crosstalk between the two cancer cells and the tumor microenvironment, the cells which are present in the tumor microenvironment, plays an important role in promoting the cancer proliferation, migration and lipogenesis. So, the 5-lipoxygenase inhibitors and the leukotriene receptor antagonists could be potential targets for the treatment of the cancers which are being exploited. So, in summary, we can say that from the eicosanoid signaling, COX-2 from the cyclooxygenase pathway and 5-LOX from the lipoxygenase pathway play an important role in the tumor promotion. And so, as you can see from this, there are various cancers in which COX-2 and 5-LOX are co-expressed and promoting the tumors, and both are pro-angiogenic. COX-2 products, specifically prostaglandin E2, and 5-lipoxygenase products, leukotriene B4, are pro-angiogenic. They in fact promote vascular endothelial growth factor, and both also are involved in arresting apoptosis. I told you that apoptosis, down regulation of apoptosis is also one of the characteristic feature of the hallmarks of the cancer, and cancer cells somehow come up with a mechanism to down regulate the expression, down regulate apoptosis process, and one of that mediators is the 5-lipoxygenase and COX-2, which down regulate apoptosis and thus promote the cell proliferation. As we have seen, the various hallmarks of cancer will form the targets for development of anti-cancer drugs, and in this chapter we have seen the role of eicosanoid signaling. So, this slide summarizes in fact the entire eicosanoid signaling pathways. As I told in the beginning that by incorporation of one atom of oxygen through monooxygenase pathway, incorporating one molecule of oxygen through the lipoxygenase pathway, incorporating two molecules of oxygen through COX pathway, generates a variety of eicosanoids, and these eicosanoids have a role in promoting cancer process. And as a result, the inhibitors of these pathways, we have discussed the COX and LOX pathways, but the other pathways also play an important role. And the inhibitors of these pathways could form the potential candidates for developing anti-cancer drugs. So, in conclusion, inflammation is a cellular immune and metabolic response to injury or infection. It is a complex interplay of cellular particulate matters designed to eliminate the cause of injury or infection. While acute inflammation is a protective mechanism, the chronic inflammation is associated with the onset of several disorders including cancer, and inflammation is a critical component in the tumor microenvironment, which is promoting angiogenesis, metastasis, and even drug resistance also. So, among various factors, eicosanoids, the oxygenated metabolites of PUFAs such as arachidonic acid formed via COX and LOX pathways are the key mediators of inflammation. As a result, COX and LOX pathways have become the targets not only for the development of drugs for various inflammatory disorders, but also for various cancers. And their role in overcoming the drug resistance is also explained, and as a result they play a much greater role in targeting cancers, in the treatment of cancers. So, in conclusion, we can say that inflammation is a cellular immune and metabolic response to injury and infection. It is a complex interplay of cellular particulate mediators designed to eliminate the cause of injury or infection. While acute inflammation is a protective mechanism to us, and the chronic inflammation is associated with the onset of various disorders including cancer. Inflammation is a critical component of tumor progression by promoting angiogenesis, metastasis, and even drug resistance, and this is all mediated by the inflammatory cells which are infiltrated into the tumor microenvironment. Among various factors, eicosanoids, the oxygenated metabolites of polyunsaturated fatty acids such as arachidonic acid formed via the LOX and COX pathways are the key mediators of inflammation, and as a result COX and LOX pathways have become the targets not only for the development of drugs of various inflammatory disorders, but also various cancers. So, our recent studies also have demonstrated the potential application of anti-inflammatory drugs in overcoming drug resistance. So, as I mentioned in the very beginning, that understanding the disease process is very critical for the development of cancer. The hallmarks of cancer helped in identifying the several targets, and here we have seen now inflammation which has been taken up as a target, and anti-inflammatory drugs are helping in the treatment of cancer patients. So, that is all for this module on inflammation and its role in cancer and eicosanoid signaling as a target for anti-cancer drug development. Thank you.